Thank you so much for joining us. Thank you so much for joining us this afternoon for our special webinar series related to the PACE project. I just wanted to introduce our speaker today, Dr. Joseph Wiemels. I hope I said that right. He is a professor in the Center for Genetic Epidemiology in the Department of Population and Public Health Sciences at the University of Southern California, and he studies the molecular epidemiology of childhood cancers, particularly childhood leukemia and brain cancer. He received his doctorate in environmental health sciences at UC Berkeley, examining the metabolism and toxicity of benzene and butadiene, followed by a postdoc at the Institute of Cancer Research in the UK, studying the molecular genetics and natural history of childhood acute leukemia and myeloid leukemia. And he's been a faculty member for over 24 years at UCSF and USC, really focusing on etiology and prevention of childhood cancers. And so it's my great pleasure to welcome him here today, and I'm so excited to hear him speak. I'll let you get started.
Okay, thank you. Thank you for that great introduction, and all of you at POCASE and around the country. So today I know this is a environmental health group, and I work on environmental health. I also work on genetics, and I also try to bring everything back to biology. And so I'm going to talk quite a bit about developmental biology in a way, and also infectious disease as causes of childhood cancer. And I don't have any financial relationship to anything I do. Okay, and just to set the stage, childhood cancer rates are increasing, and they've been increasing, a little less than 1% per year, and this is overall, including leukemia, brain cancer, and all of the more rare cancers. And the main point of this graph is to show that yes, there's some environmental, there has to be an environmental cause to this, because our genetics as human beings has not changed over this time period so quickly. Now, when I talk about environment, I'm not only talking about chemical exposures that we've created and exposed ourselves to over time, but also diet, the way that we live, the way that we interact with each other. Those are all going to be have some impact on why these rates have been increasing. So in my research, most of my effort has been on the two most common cancers in childhood, leukemia being the most common. And leukemia has also had this increase over time. It's been about 70% since the end of World War II. And most recently, the group where rates are increasing is Hispanic, and Hispanics or Latinos, which we still see this 1% per year, or even higher than 1% per year, although it seems to have flattened out in other race ethnicity groups. So the two parts of my talk today, I want to focus on two time periods in the life of the child. One is what's going on in early gestation in the womb, in early and also late gestation. And then again, after birth, when we look at the interaction of children with their infections and the immune stimulants in their early life. So mild infections, fulminant infections, also vaccines and specific infections. First of all, we'll look at what's going on before birth. So years ago, we went and looked at genetic changes that are evident in leukemias, and we found that some genetic changes are definitively happen before birth. And this is from an early study where we looked at twins, where two twins both got leukemia. And let me see if I can get my pointer. Do you guys see a pointer? Is this it? Laser pointer, here it is. Okay, so this is twin A and this is twin B. We can see that they share this band here, that in this gel over here. That band refers to this sequence that doesn't exist in the normal human genome, but it's a translocation between a gene on chromosome 12 and 21. So if they share that mutation in the diagnosis, and one child got diagnosed at age 5 and one child got diagnosed at age 13, they have the exact same translocation. That means they most likely had shared that even before they were born, even though it took, in the case of the older one, 13 years for it to finally result in a leukemia. And we've also found that this is not something only with twin children, but we've looked at other, the same translocation there, the ETV6-RNX1, plus other ones, and we found that they also commonly occur prenatally. And then we and other research groups have looked at a host of other genetic events and found that there are some that reproducibly happen after birth, such as gene deletions and mutations in the RAS pathway. So this really firms up the idea that there's early hits and later hits in leukemia. But these translocations, they occur in hematopoietic cells, and there's some thought that there could be other events that happen even earlier in the gestational period. And I'll show you some results also from twin studies where we looked at twins that in this case, they didn't both get leukemia. Only one got leukemia and one did not. And this is far more common. This scenario is far more common. So identical twins where one gets leukemia and the other one stays healthy. So we wanted to see, is there some difference, even though these children are genetically identical, is there some difference that we can detect at birth? And sure enough, there is a difference in DNA methylation, which is what we're looking at in this figure here. So DNA methylation is what we call epigenetics. It's above the gene where methyl groups are attached to the promoters of genes to permit expression. They're also attached to the backbone of the genome to make the genome stable and not exhibit instability and genetic mutations. What we found is that in this set of 43 twins that we looked at was that there was an under-methylation or a hypomethylation in the twin that got leukemia compared to its control, reproducibly over the set of twins. And when we looked at where this DNA methylation difference occurred, it was not in gene promoters. So gene promoters, or the control region of the gene, which is over here, the promoter and the CPG island near the gene. Instead, the DNA methylation difference was in the open C, or the part of the genome that does not have a lot of genes. And this indicates that this open C region may become hypomethylated in one twin, making the genome more unstable and liable to having genetic mutations later on. So what could it be? We know that these twins are genetically identical, and what difference could there be? I mean, they're in the same mother, they're born on the same, they have the same birth date, they have the same gestational time inside of mother. There can be some differences between the twins. The umbilical cords can be more functional for one twin versus the other one, so there might be nutritional differences. Sometimes there can be infections that aren't shared, just one child gets it versus the other. But recently we had another observation with twins, which is quite curious, and it's something we can't quite explain, but we have some initial ideas, and we can discuss later if anybody has more ideas. But what we found is that when twins when one child gets lymphoid leukemia and the other one does not, so this is from California registry data. So the second child here is much less likely to get leukemia if only one child gets leukemia. So it's it's almost a 2-to-1 ratio where the first one gets leukemia, the second one does not. So this may be that there's some protection from getting leukemia in the second one out in a twin birth. What could that be? Well, is it because of C-section versus natural birth? It turns out we'd see the same ratio in both of those groups. But interestingly, we find that the same CpGs we know vary by birth order in singleton twins, also vary in when we compare identical twins at birth. We also see that cortisol responsive CpGs are also significantly different in the first twin versus the second twin. And the reason we looked at this was this idea that stress-induced cortisol may actually be protective against leukemia. And the reason that hypothesis was brought up a few years ago by Kjeld Schmigelo from Denmark, what he did is he looked at the fact that children who have had elective C-sections have a higher risk to leukemia, and the thought was that maybe the stress of birth is actually toxic to leukemia cells because some of these cortisol is related. It's a steroid related to prednisone, dexamethasone, which are drugs that we use that are highly effective against leukemia. So oddly, it may be in this case that stresses may be anti-leukemic in this scenario. Although we don't, this is still a hypothesis at this point, and if anybody has other ideas, please let me know. This ratio we see with lymphoid leukemia here has now been replicated in another 5-state study, unpublished data, but this is clearly a bona fide result. So we really want to help try to explain what might be going on here because it may have implications for leukemia prevention. So switching back to folate, and the reason for doing that is our initial thoughts with the twins was that it had something nutritionally involved. We're not sure about that now, but we know in singleton births that nutrition is a strong risk modifier for leukemia based on, this is in our California childhood leukemia study, again, where we see that high intake of vitamin supplements or simply taking vitamin supplements of prenatal strength is protective against leukemia, especially in Latino mothers. Now, more recently, we worked with a group from France and from Norway and also Australia, which also saw the same profile in this gene when we compared children at birth who grew up to get leukemia compared to controls. And we found that this gene VTRNA2-1 DNA methylation was lower in cases compared to controls. Oh, sorry, the other way around. Higher in cases compared to controls. And what does this have to do with folate? Well, it turns out that this gene is a very strong marker for nutrition, nutritional differences before birth. And this comes from a study from the Gambia in Africa, where seasonal nutrition is quite varied. What's available during different seasons. And what this group found was that children who had early gestation during the rainy season compared to the dry season, The rainy season, there are not a lot of fresh vegetables available, but then they ripen during the dry season. So that's the harvest season, where the available foods are much more diverse and healthy. And there was this gene was the clear winner in this study for being different in a genome-wide analysis between children who had early gestation in rainy versus dry season. Now, in California, we also have a rainy and a dry season, but it doesn't really alter the available foods. We pretty much have healthy foods available all year round. But what we find is that children who grow up to be leukemia cases, look a lot like the children who had early gestation during the rainy season in Gambia, indicating that those mothers may choose to eat differently in early pregnancy when DNA methylation is established at this gene. So this mirrors what we're seeing with folate being protective for leukemia. We see this VTRNA2-1 as being kind of a biomarker of that. So if we have a reduction in methyl donor foods or healthy foods that have folate and B vitamins, this leads to an increase in DNA methylation in this VTRNA2-1, which both of these are associated with risk of childhood leukemia. We're not exactly sure if VTRNA2-1 is a leukemia-inducing gene, but at least we know that it's a biomarker of risk. Now, how are we going to deal with this in the population? Well, we already tell women or tell families to eat well and to take prenatal vitamins before you're pregnant or before you're planning to become pregnant. It really is those critical first few days of pregnancy where this DNA methylation is set up. But are some women more susceptible than others to folate deficiency leading to leukemia risk? Well, what we did in this analysis here is we took genes or genetic SNPs, which are associated with folate in very large studies. So there's a large meta-analysis here, this Van Moor study, which looked at about around 45,000 people where B vitamins were measured in the blood of those individuals. So they measured folate, B6, B12, and homocysteine. And you can see where these vitamins are in the folate pathway or the 1-carbon pathway. We have methyltransferases over here, which is where DNA methylation of the genome, those methyl groups come from. We also have nucleic acid synthesis over here. So you can see if you're limiting in any of these vitamins, you may end up slowing or altering this pathway and resulting in less efficient DNA methylation and other cellular processes. So the SNPs that predict for folate, you can see that those SNPs, if you if you have SNPs that will predict for higher folate, that will lead to reduced risk of leukemia. Just like when we ask individuals what they eat to do a food frequency questionnaire, folate also reduces risk of leukemia. So it turns out that genetic prediction of folate also reduces risk. Genetic prediction of homocysteine slightly increases risk. And this would make sense. Homocysteine is a toxic metabolite. It's also involved in heart disease or blood vessel pathologies. And we also see that there may be an increased risk with excess vitamin B12. Not much going on with vitamin B6. But what Yi Jin, who did this analysis, what she was also able to see was that people could overcome their genetic deficiency of folate by having by having adequate intake or high intake of folate. So this is in our California childhood leukemia study again, and we can see among Latinos, these are, if we look at polygenic risk score for folate levels on this axis, on the y-axis. So this is your genetic prediction of folate. If your genetic prediction is low and you also have low folate, then you end up with an increased risk of leukemia. But those individuals who who have adequate or high levels of folate, they are able to overcome this genetic deficiency. And over here we have a similar thing with homocysteine. Here, homocysteine is a risk factor rather than a protective factor, and you're able to overcome this higher levels of homocysteine if you intake more folate from food. So this is the public health impact of this observation is that if women or families know that they have a genetic liability to low folate levels in their blood, that they would be able to overcome that with adequate folate intake. Now, there are some classic animal experiments with mice that are engineered to react with folate intake in specific ways. This is a classic mouse experiment where a particular what's called an agouti allele will become expressed if when mice are deficient or when their levels of folate have been varied in their diet. So we can see that when we supplement different levels of folate in this mouse model, they have this observable coat color difference. But what's also interesting is that there are other genes that are folate impactful that will result in a different phenotype. So the ones that are yellow end up being much heavier or more obese and more likely to be impacted by other diseases such as diabetes. Whereas the ones that have their agouti allele expressed over here based on folate modification, there are other genes that are also impacted that will create a more lean mouse. And this is another example of a similar gene which is can be switched on and off. And in this study, they didn't use folate or any environmental variables, but I think it may be a mouse model that will be used for these type of experiments in the future, where this mouse with this TRIM28 mutation, their litters will have about 2/3 of them will have TRIM28 turned on, and about 1/3 of them will have TRIM28 turned off. And this is a stochastic random distribution. So even though these mice are all genetically exactly identical, they end up with the litter of having some heavy, some light mice. And the heavy mice end up having, in a more recent study where they looked at cancer in these mice, that they end up with a different network or a different array of cancers compared to the ones that have the TRIM28 on. You can see they end up with cancers in several organs, whereas the ones with TRIM28 off only have a couple of cancers here, the prostate and the esophageal cancer. And when they put it in the presence of other genes that are known to be cancer predisposition genes, in this case the TP53 gene, they will even have another variety of cancer differences between the two TRIM28 phenotypes. So this should have some impact in humans, we are all developing down a pathway which is somewhat random, which can be pushed one way or the other by environmental exposures, by dietary exposures, and these animal studies should be valuable, and at least now they're indicative of this process, which we really haven't figured out exactly how to look at in epidemiology studies, but that's for the future. Now, we've talked a lot about diet and folate and maybe this birth order effect, but are there other exposures prior to birth that might impact developmental trajectories leading to cancer risk? Yeah, I think the short answer is yes, but I'd like to just show one, which is from a recent study published this last year, which really focuses on this time period of life, being the first few days after fertilization, as being particularly critical for environmental exposure and cancer risk. This is a study also in California where ambient temperature was assessed in relationship to children getting leukemia in early life. And in this study, ambient temperature, which was described as being 5 degrees or more in a certain period during the months of May to September only. So we didn't look at the winter months because having 5 degrees extra doesn't mean a whole lot, and it doesn't exactly mean heat stress, such as right now. If it's 5 degrees more in the middle of February, we're pretty happy. But so we didn't look at the winter months. But during the summer months, there was only one period in development which there seemed to be an increase in risk of leukemia in the offspring. And we also looked at the child during its early life as well. So the heat stress on the child after birth had no effect on leukemia risk. The heat stress only had this effect during the first few gestational weeks. So this is this really peaks at week 8 after what we can predict as being the conception date of the child. So to kind of sum up this part of the talk, we really see that it's this early time period, most likely just the first few days when we set up the genome for epigenetic in epigenetic development, where a window of susceptibility to diet and environmental factors should play the largest role, and seems to play the largest role when we look at diet, when we look at heat stress during pregnancy. So just to sum up where I am now in this talk, today's talk, the part 1 is really looking at epigenetic impact, where developmental processes can lead to later cancer risk. And that we think is very early in pregnancy. At the beginning of the talk, I talked about translocations that lead to that occur in the children with leukemia, and they happen during later gestation. And about 1% to 5% of children are born with these genetic translocations. Only 0.1% of those will end up getting childhood leukemia. So what we call the second hit that then leads to the full-blown disease. So in the second part of my talk, I'd like to talk about infection as being really one of the main modifiable risk factors that we see as being important for this second hit, and also manipulating infection or response to infection may be a way of preventing leukemia in the future. So when we talk about infection, there's various ways to measure infection. One of the earliest and one variable that there's a lot of data out there is looking at early exposure to infections through daycare attendance. So simply quantifying how long children or at what age they go into daycare, how many other children are in the daycare. And what these studies show in some is that more exposure to daycare seems to be protective. So simply being exposed to other children leads to a lower risk of leukemia. However, there's one kind of paradox here, and I'll take this one study as an example. This is Gillum 2005 study. This is the United Kingdom Childhood Cancer Study. It's one of the biggest ones in this meta-analysis here. What they showed, even though that more exposure to daycare was protective in their children to leukemia risk, they saw that children were actually in the doctor's office more often, the ones who got leukemia. So it looks a little bit like a paradox, because if they're in daycare, if daycare is lowering their risk, isn't daycare also increasing their exposure to infections? So it seems a bit like a paradox, but I think another way of looking at this is the way that children are dealing with the infection. So not simply being exposed to infection, but actually getting sick enough to being taken to the doctor. That's a risk factor, whereas being exposed to infections seems to be protective. And this was another study that we did that kind of confirmed the same thing. So we see higher odds ratios, the more number of clinic visits for infection. This is in Taiwan, where they have a national healthcare where you can do this kind of work just with data records. And so you can see it's quite a strong risk from having fulminant infections that may put you in the doctor's office. And so if we try to kind of summarize these exposures, we see that early antigen exposure we get through a situation like a daycare leads to trained immunity and immune competence and health, whereas delayed antigen exposure, for instance, if you didn't go to daycare at all and you got all your infections during, when you ended up in school as a kindergartner, that's related to potentially dysregulated immune responses that are too strong and damaging, and will then cause expansion of pre-leukemic clones. So if this sounds like a familiar scenario to other diseases, it certainly is. This is the same scenario as the hygiene hypothesis, which is proposed as being a way that children can be healthy in relationship to asthma and allergies. Getting early exposure is a good thing to a wide variety of antigens, whereas having an environment too clean leads to higher risk of asthma and allergies. Now, I think pertinent to to this point in time where there's changes in our government, and there talks about whether vaccinations might be damaging. Let's take a look at that over the next few slides. So we know that vaccines stimulate the immune system. So the question is this stimulation going to be gentle modulating type stimulation, like the common infections you get in daycare, or will vaccines cause damage and increase to leukemia? And as healthcare professionals, we are probably going to get asked these sorts of questions. So can vaccines cause cancer? There has certainly long been a scare that this would be true, in part because of the early polio virus vaccine that was produced in monkey kidney cell cultures. In the early years of that vaccine, those vaccines were contaminated with SV40, which is a simian virus. And this virus contains what's called a large T antigen, which can inactivate tumor suppressor genes like TP53 and RB. And it has some similar structures to some other viruses that occur in humans, such as BK and JC viruses. So investigators have long looked for presence of SV40 in human cancers to see if these poliovirus vaccines may have impacted cancer risk. And they have found them. So they have found these sequences in certain cancers. But really, there is no definitive epidemiologic evidence when we look at the people who have had those early polioviruses versus ones that came later after 1963. There really doesn't seem to be a strong cancer or any cancer risk. The whole vaccine, kind of the idea that vaccines are harmful in more recent years came out of I'm sure you're familiar with the report from the late 90s about potential link to autism. But just to reiterate there was no connection there. The original report was falsified, and the person who made the report themselves was trying to sell a test for make millions of dollars off this. He ended up losing his medical license, and nothing was proven in the years after that. Now, do we have any modern viruses or vaccines that have been introduced recently where we can look at risk to leukemia? There's really only one that was kind of introduced over the period of time when people have been actively doing case control studies on leukemia. And that's this Haemophilus influenza type B vaccine. And a couple of reports, one from Finland, who looked at a very large trial and found that getting the HIB vaccine, it turned out to be those children who got it had a lower risk to leukemia, interestingly enough. So not that the virus is potentially causative, but it may be doing this type of immune stimulation, which is healthy, sort of a mild immune stimulation. And we saw the same thing in a California study here. Because HIB was introduced over the time period of our study, we could compare people who had the vaccine versus who did not. We were not able to do that with any other vaccines because pretty much everybody got every other vaccine at the same time in normal vaccination schedules, so we didn't have a control group, but we did for HIB. So here's a summary of vaccine results in childhood ALL. So the HIB ones are up here on the top. You can see that those were introduced over the last few decades, where we can see a reduced risk. Other investigators have tried to look at vaccines, and you can see that the result is kind of all over the place. Some of these studies are very small, and some of them were not well controlled for confounders. But there is a very nice recent study here, which is from the Danish, their registry, which is, pretty much covers the entire population. So we're looking at, in this case, over about a 10-year period, over 1 million children with 409 who got ALL. And in this study, you can see there really isn't any association with vaccination and subsequent leukemia. So I think this may be the most definitive or the largest study, at least, showing a lack of association, which I think is kind of the message that we would like to give to people who ask. So in the final part of this talk, I'll talk a bit about whether we know that children have these predisposing epigenetic and genetic changes by the time that they're born. But we asked a few years ago, is there anything about the immune system that's different in children who get ALL in addition to those other other genetic and epigenetic events that I talked about earlier. And we're able to use these population-based resources in California, where we can investigate immune status at birth through the neonatal blood spots. And in our California childhood leukemia study, we've also linked those up with the tumors after birth. So we started this more than 10 years, actually 15 years ago, looking at cytokines at birth in children who grow up to get leukemia, and found that there was one cytokine which was vastly, vastly different, profoundly repressed in children who went on to get leukemia. That's this cytokine interleukin-10, which doesn't really have anything to do with the genetic changes we talked about before or the epigenetic changes. It's really something quite distinct. So we asked what causes this profound interleukin-10 deficit, and we thought maybe it had something to do with an infection. So we looked for an infection that might be impactful in lymphocytic leukemias. We looked at diagnostic leukemia DNA, as well as isolated viral particles from leukemia patients. And we compared lymphocytic leukemias with myeloid leukemias. And this is the first time that a research group that I'm aware of used another set of sick children to investigate whether a virus is involved in leukemia. And the reason for using AML, we don't have any infection epidemiology for AML like we do for ALL. And we know that these children are also in the clinic, just like the ALL kids are. They have suppressed immune systems. So we would expect them to get to have visible infections at the time of diagnosis, which they did. But what was interesting, though, is there was one infection which distinguished lymphocytic leukemias from the myeloids, and that's cytomegalovirus. So CMV here in RNA pools from lymphocytic leukemias, we always saw cytomegalovirus, and we didn't see it at all in AMLs, even though the children are on average older for AMLs, and they have more opportunity to contract or be exposed to CMV. They didn't show any at diagnosis, at least in this initial data set here. And wondering why CMV, what time did they get it? We looked at neonatal blood spots and found that children were much more likely to have CMV at birth than healthy controls. And there was no difference with Epstein-Barr virus, which is another virus known to be involved in hematologic malignancies. So it really pointed the finger more directly at CMV being the potential cause. And this was replicated in a Swedish study where CMV disease itself was looked at in relationship to leukemia. All these hazard ratios are above 1, quite a bit above 1. In fact, overall about an 11-fold risk if you have CMV in the mother or at birth in the child. So what might be, how does this relate to that interleukin-10 result that I showed before? Well, we tried to look at CMV in relationship to neonatal cytokines. And here we looked at CMV IL-10, which is a version of IL-10 that's encoded by the CMV virus itself. And we wanted to see if this was present at birth in children who get leukemia compared to controls. We didn't actually see a big difference, but we were able to show some significant correlations with maternal cytokines in mid-pregnancy in this study here. So the idea here is that we thought that CMV IL-10, which is this viral version of IL-10, may be pushing having some kind of an effect on human IL-10, and that's why it was so low at birth. I should say we haven't quite gotten the evidence to determine that is actually happening, in part because these cytokines are so they're so ethereal, or they degrade very rapidly in these neonatal blood spots, and it's hard to get the blood spots in time to do these particular analyses. But at least this is the hypothesis that we're working with now. And this has obviously profound implications for leukemia prevention. If we can prevent CMV infection from mother to child, either during pregnancy or around the time of birth, then we may be able to prevent a significant portion of lymphocytic leukemias. More to delineate a role for CMV in leukemias, we reported a couple of other interesting observations. One is that if we look at children with high levels of CMV in their diagnostic sample, we see that they have a lot higher report of self-reported infections after birth, indicating that this the CMV may somehow be impacting their immune function and making them more liable to damaging infections in early childhood. We also see differences in CMV levels by different leukemia subtypes, including the high hyperdiploids are much more likely to be CMV positive and have high levels of CMV, leading to this scenario here, where we found that if children are exposed to CMV in utero or early infancy, these are the children which then display more evidence of damaging infections leading to leukemia. And this is the non-high hyperdiploid leukemia. Interestingly, the leukemias that have the highest levels of CMV are the high hyperdiploid leukemias. They don't show this high infection, this strong history of infections after birth. So there may be different means by which CMV influences leukemogenesis. Okay, just as a summary here, these are the many different exposures that pertain to mostly to immune modulating risk modifiers. We start out with genetic and epigenetic alterations very early. Now, C-section is associated with increased risk, CMV increased risk. Being a firstborn is increased risk. That's true for both singletons and twins. Having cytokine disruptions at birth, breastfeeding decreases risk of childhood leukemia, daycare attendance decreases, and finally, childhood infections or the fulminant infections that puts children into the doctor's office increase risk. So for me, when we look at infectious exposures and how they impact leukemogenesis, I always come back to this interleukin-10 observation, which is so profoundly different in children who grow up to get leukemia compared to controls, that really that's still my center point of thought when we look at all the impacts that potentially will influence neonatal IL-10 levels. When you end up with that low level of IL-10, you have an immune system that responds to infections in an aberrant manner. So that low IL-10 will lead to lack of suppression and increase risk of childhood leukemia. And if your normal levels will then permit you to respond more with an attenuated response. So it really is that responder status at birth, which is critical. Now, what I haven't talked a lot and what's not in this picture is the effect of environmental or chemical exposures. And I want you to stay tuned because I think Catherine Metier, my colleague from Berkeley, will be giving a talk on environmental exposures that will come again, that will come up this spring. And I'll leave that to her to give you all those details. But I want you to keep in mind a lot of those environmental variables also impact immune function and immune status in ways that we're just beginning to understand. And finally, some thanks to people working in the lab here, and also my colleagues from historically from UCSF and UC Berkeley and Yale. Okay, and I'd be happy to answer any questions. So feel free to type into the chat if you wish, or just turn your camera on and speak.
Well, Joe, that was just a whirlwind of a story. It's actually slightly difficult to follow every thread of it through. This story was about the role that, well, you had two. One was nutrition and how that impacts things. With folate story is like the first days after conception, right? And
Right.
the infection story, it seems complicated. There are different things going on. There's innate IL-10 levels, and there's childhood infection, and then there's CMV, which is during pregnancy, right? Or is that during pregnancy or in early childhood? And how do these all fit together? Yeah.
Yeah, how do they all fit together? Well, it definitely shows that there's a multi-step pathway to cancer, to childhood leukemia. And if any one of these steps were prevented or changed, the leukemia would not be there. So it's really got to be the concordance in one child of everything happen. We know that the pre-leukemic mutations are present in 1 to 5% of children, and so obviously 5% of children don't get cancer. So it's got to be the 5% that have the pre-leukemic mutation, but they also have early epigenetic event, and then they also have an aberrant response to infections after birth. So that all those things have to happen together to be that 1 out of whatever 300 kids who will get the cancer. So even though the mutations and the events are quite common, so what it does is it gives us several avenues of promoting health and avoiding cancer. But we definitely are talking about independent events at several times in a child's life, and infections are not going to mean anything at the time of conception because there's no immune system. But in developmental processes where folate is critical, we know from other studies like the Dutch Hunger Winter study and the Gambian study that it really is that time right around conception that's so important for nutrition. Nick, I see your hand up.
Yeah, hi. Great talk. I'm wondering, just trying to connect the two, like the folic acid and the infection risk. I mean, is there any sense about whether the methylation changes are taking places in parts of the genome that would be relevant to an immune response, or are they um, is it just like a separate? I mean, I understand they're like non-coding regions. I'm guessing from what you said, but is there any potential connection between all those things?
There's likely to be a connection. So if the work on the DNA methylation showing that the backbone of the genome has this lower methylation status, it will make the chromatin more unstable, more able to be attacked by whatever chemicals or reactive oxygen species that are created during, for instance, an infection response. So when we do have an infection response, we have inflammation, we have blood cells that release factors that kill microbes or destroy viruses or destroy virus-infected cells. And those will increase the level of these reactive oxygen species that then can cause the genetic mutations for this unstable DNA that was created very early. So there's likely to be some combination, although that's quite speculative at this point. We don't have, I would say we don't have really strong experimental evidence.
Sure. No. It was just I was just thinking through, like, you can imagine there are people who are both like nutritionally poor, like poor nutrition, and they have exposures to various infectious diseases during this post-conception time, and you know you're talking about prevention, like how could we like hedge one or the other. So yeah, thank you very much. It's really interesting.
Okay, I have another question in the chat. Is there correlation between planned pregnancies and unplanned, being that the first few days of conception are key? Well, that's a great question. I don't think we've ever asked mothers of children with cancer whether their pregnancy was planned or not. But we do ask them detailed questions about diet and whether they're taking prenatal vitamins, for instance. And we might imagine that somebody who's not taking prenatal vitamins may also not be planning on having a pregnancy. So we could maybe by proxy answer that question with that. And we do definitely see a higher risk in mothers who well, I should say we see protection from prenatal vitamins that people take before they're pregnant, in the year before they're pregnant. So certainly those would be planned pregnancies and reduce risk from taking prenatal vitamins. All right, I think we're right at time, but if there is any other questions, feel free to reach out.